सेल thawing एंड रिकवरी के बाद जो नेक्स्ट स्टेप है टू इंश्योर द लाइफ सेल्स, द काउंट ऑफ द लाइफ सेल्स, वी नीड टू परफॉर्म सेल काउंटिंग. सेल काउंटिंग के हवाले से हम पहले भी मॉड्यूल में बात कर चुके दैट वी यूज़ हीमोसाइटोमीटर फॉर मैनुअल सेल काउंट और सम टाइम्स वी हैव द ऑटोमेटेड सेल काउंटर्स अवेलेबल. जब भी हम सेल कल्चर में से फ्रोजन सेल्स को थॉ करने की तरफ जाएंगे, देन वी हैव टू इंश्योर दैट व्हाट पॉपुलेशन ऑफ सेल्स आर स्टिल अलाइव एंड व्हाट पॉपुलेशन is no more alive. For that purpose, using hemocytometer, we perform cell count. And if you look at the picture of the hemocytometer, the purpose of this chambers is to identify or is to perform the cell count. If the number of cells or the cell density in the given sample is very high, we use a different area. If we have not very high cell density, and we can use a different area for cell counting. Hemocytometer was basically designed to count the blood cells, for red blood cells and for white blood cells. जो central area है इस hemocytometer में that was used or that was purposefully designed for the count of red blood cells because of large number of these cells in the blood, and the white blood cells were counted using the four corners of that particular hemocytometer. अब हमारे पास जो cells हैं for counting, we determine the cell count using either of the area, whether that particular area designated for red blood cell or that particular area designated for white blood cells. इस पिक्चर में अगर देखें आपको 1, 2, 3, 4 और 5 के हवाले से कुछ एरियाज विजिबल हैं. The area number 1, 2, 4 and 5. These areas are basically meant for white blood cell count, and area number 3 is meant for red blood cell count. We can use either of the region, and keeping in view the principle of cell counting which we adopted for white blood cell count or red blood cell count. मैं step wise detail में नहीं जाऊंगा इस procedure की, because we already have discussed how to use it and how to count the cells after diluting the sample. Materials which are required for performing the cell count: cell suspension is required, the balanced salt solution is required, then we need a stain which is known as trypan blue stain. We use 0.4% of trypan blue. ये ready to use stain available है market में, commercial sources से मिल सकता है. इस stain की एक property है that it stains the dead cells as blue. अब हमारा पर्पस इस काउंट का ये है कि हम आइडेंटिफाई कर सकें लाइव सेल्स और डेड सेल्स, व्हाट इज अ काउंट एंड व्हाट इज अ रेशियो. फॉर दैट पर्पस व्हेन वी मिक्स द सेल पॉपुलेशन, बैलेंस्ड सॉल्ट सॉल्यूशन and we mix the trypan blue, let it settle on the bench for 5 minutes. In 5 minutes में what is going to happen, जो trypan blue है, it will enter through the plasma membrane into the cells. अब जब हम 5 मिनट की इंक्यूबेशन के बाद, 5 मिनट की सेटलमेंट के बाद सेल्स को हेमोसाइटोमीटर में लोड करते हैं, तो हमें दो किस्म के सेल्स विजिबल होते हैं. दो सेल्स which are stained blue and the other cells which are unstained. The unstained cells are live cells. The blue cells are dead cells. So we have to make a count of both blue cells as well as white cells. Blue cells will give us a total number of dead cells. White cells will give us a total number of live cells. Keeping in view these numbers, we can find out the percentage of viable cells, and then we will have an idea that how many cells are present in that particular vial which are alive, and what number or what volume we need to take from that particular vial to establish our culture again.